Chemical composition and antimicrobial activities of the oil. Essential oil of Mentha Rotundifolia. Abstract essential oils extracted from aromatic or medicinal plants have recently proved different applications in various fields, manufacture of medicines, perfumes, and foodstuffs. The objective of the study is to evaluate the antimicrobial effect of essential oils extracted from the plant Mentha rotundifolia against pathogenic strains. Essential oils are extracted by the Clevenger hydrodistillation technique. The results obtained show that the yield of the essential oils is of the order of 1.58% and the chemical composition of the essential oils is determined by the gas chromatography (GC) technique. The products obtained with their proportion are 2-isopropylidenecyclohexanone, 11.99%, eucarvone, 11.42%, gamma-muoroline, 8.61%, 2-isopropyl-5-methyl-3-cyclohexane 6.83%, and p-mentane 1,2,3-triol, 6.72%. For antibacterial activity, the essential oil of Mentha rotundifolia is shown to be effective against bacteria, Portus mirabilis, susceptible with a diameter of 5, 6, Acinetobacter baumannii, sensitive with a diameter of 1.6, Salmonella suci, susceptible with a diameter of 1.4, Escherichia coli, sensitive with a diameter of 1.2, Klebsiella pneumoniae, sensitive with a diameter of 1.1, Citrobacter freundii, intermediate with a diameter of 0.8, and Staphylococcus aureus, 1 cm. While the bacterium Pseudomonas aeruginosa, 0 cm, showed resistance against the inhibitory effect of essential oils of Mentha rotundifolia. Keyword: essential oils, Mentha rotundifolia, pathogenic strains, hydrodistillation. Introduction. Mentha species are grown in several countries around the world for the production of essential oils. Sudur et al., 2010, Lajal et al., 2011. According to the flora of Algeria, this genus is represented by 5 main species: Mentha rotundifolia, Mentha longifolia, Mentha spicata, Mentha aquatica, and Mentha pelagium, Quesel and Santa, 1962. M. rotundifolia, L. Huds, Mr., commonly called green apple, is a perennial herb with wild growth. It is widely distributed in northern Algeria in the subhumid areas, along the rivers in the plains and mountains where it is known as Timija or Timarsat, Brada et al., 2006, and it is widely used. For example, a leaf decoction is made for topical application to treat furunculosis and abscesses, to reduce fever and as a mouthwash for dental pain, Brahmi et al., 2014b. In addition, the plant would treat bronchitis, cough, and ulcerative colitis. It is also taken as a tonic, used as a stimulant, stomach, carminative, analgesic, choleretic, antispasmodic, sedative, and hypotensive, as well as a common spice, Lajal et al., 2011. This oxygenated monoterpene has very interesting biological effects. It presents cardiovascular effects, hypotensive activity, vasodilator, bradycardia, activity on the sympathetic nerve centers, relaxant, stimulant, depressant, antibacterial, and antifungal properties, and also acts as an agent delaying the reproduction of the malaria vector Anopheles stephensis, Damian et al., 2003, Tripathi et al., 2004. Piperonidine oxide is also of interest for the synthesis of heterocycles, pyrosols, pyrosolins and allylic alcohols, Golomai et al., 2001. The composition of essential oils among species of the genus Mentha has demonstrated a chemical diversity due to geographical environmental factors. Beghija et al., 2007, Brada et al., 2007, Ouhssine et al., 2010, Kumar et al., 2011, Baser et al., 2012, Sitzman et al., 2014, Kasrati et al., 2015. A research in the literature reveals some studies on the biology activities of MPE and MRE conducted on plants harvested outside Algeria. The importance of the essential oils of the plant M. rotundifolia is illustrated in many scientific works, describing the chemical composition of the plant, contain the following compounds: transpiperitone epoxide with a percentage of 30.2% and piperitone oxide with a percentage 8.7%. F. Brahmi et al., Industrial Crops and Products XXX, 2016. The objective of this work is to study the antimicrobial effect of the essential oils extracted from the leaves of Mentha Rotundifolia grown in the region of Al-Tazakanitra province on pathogenic germs from our microtheca and to determine the chemical composition of Mentha Rotundifolia essential oils, growing wild in this region and highlighting a possible variability in the chemical composition of these oils. This study is part of the broader context of enhancing the biodiversity of Moroccan aromatic plants for their medicinal and food properties. 2 Materials and methods. A plant material. The harvest of the plant was carried out between March and September while its flowering is between June and August. Only the aerial part, leaves, was dried in the open air, in the shade and at room temperature, 25 degrees Celsius. B. Hydrodistillation. Extraction of the essential oil was carried out by hydrodistillation in a Clevenger type apparatus. The plant, once dried, is decorticated with its leaves and then put into the 1-liter flask with a quantity of 100 g added with 500 ml distilled water. The whole is boiled for a period of 3 hours. 3 distillations were performed to recover a volume of 1 ml of the essential oil. The recovery of essential oils is made by cooling the refracting material. The resulting extract is dissolved in ethanol solvent and placed in a cryotube and stored in the dark in a refrigerator at 4 degrees Celsius 21. See Microorganisms Studied. The bacteria studied are, Portus mirabilis, Acinetobacter baumannii, Salmonella typhi, Escherichia coli, Klebsiella pneumoniae, Citrobacter freundii, Staph aureus, and Pseudomonas aeruginosa were chosen for their high frequencies to contaminate foodstuffs and for their pathogenicity, and the bacteria tested are cultured on their specific media for their recovery. The minimal inhibitory concentrations, MIC, of the essential oil of Mentha Rotundifolia are determined according to the method reported by Rhaim et al., 1993, and Farah et al., 200i.d for their pathogenicity. A determination of the minimum inhibitory concentration. The determination of the minimum inhibitory concentration, MIC, of the essential oils was carried out according to the method reported by Rhaim et al. And Saitrani et al. Due to the immiscibility of the essential oil with water and therefore with the culture media, emulsification was carried out using a 0.2% agar solution in order to promote the seed/compound contact. In order to find the minimum inhibitory concentration of essential oils, a series of dilutions of this oil is prepared as follows: 1/10, 1/25, 1/50, 1/100.1/200, 1/300 and 1/500 in an agar solution. In test tubes, each containing 13.5 ml of Mueller solid medium, are added 1.5 ml of each of the dilutions so as to obtain the lowest final concentrations of the essential oil which inhibits any visible bacterial growth. Naked eye including 1/10, 1/25.1/500, 1/50, 1/100, 1/200, 1/1300, 1/500, v/v, then the contents of each tube is poured into a petri dish after stirring. Controls, containing the culture medium and the 0.2% agar solution, are also prepared after the incubation time, 16 to 20 hours, at 37 degrees Celsius. B. Determination of the minimum bactericidal concentration, MBC, is determined following the inoculation of a sample of the non-growth plates on Mueller-Hinton agar. The lowest concentration of essential oil resulting in the death of 99.99% of the bacteria after incubation at 37 degrees Celsius corresponds to the minimum bactericidal concentration CMB. Each test was repeated 3 times for confirmation. D. Gas chromatography, GC. This is a separation technique with an apparatus that contains a column exists in the oven consists of separated volatile constituents in a mixture. Their use depends on 3 parameters that are, sensitivity, reproducibility, lifetime. We begin by introducing the sample which takes place in 3 phases. Spray of the sample. Transfer of the sample to the column. Initialization of the gas chromatography, GC, oven program. GC mechanism. Analytes cross the column. They are separated according to 2 criteria, volatility and polarity. Separation of compounds. As the elution progresses, the compounds are quantified in the detector. The retention time, TR, is the passage time of an analyte in the column. The chromatogram is the representation of the intensity in the detector during the retention time, table point 1. Here results and discussion. A. Chemical composition of essential oils. The results of the analysis of the essential oil extracted from the plant Mentha Rotundifolia are grouped in the following table showed the presence of chemical substances. The study of the literature reveals differences in the qualitative and especially quantitative composition of the essential oil Mentha Rotundifolia. Indeed, we have seen that the contents of the different compounds are far from fixed and sometimes vary a lot from one study to the next. In order to compare the results obtained and to try to understand these variations in Mentha Rotundifolia L. Climatic conditions. The age of the plant. The harvest period. Desiccation. Conservation of the plant. Analysis of the chemical composition of the essential oil, derived from the plant Mentha Rotundifolia, grown in the region of Sidi al-Taza province of Kenitra. By gas chromatography, showed us the presence of the two dominant elements, 2-isopropylidenecyclohexanone with a percentage of 11.99% and eucarvone with a rate of 11.42%. In comparison, these percentages are totally different from those of the essential oil from the same plant grown in Milyana, which is located in the south of Dara, whose major compounds are piperitone oxides. 31.4% piperitone oxide has been reported as the major constituent of the essential oil of M. rotundifolia from Greece, Kokanai, Pape Georgiou, 1988, and Germany, Van O.S., Hendricks, 1975. The variation of the chemical composition of the essential oils of M. rotundifolia N revealed the existence of particular chemotypes with as major compounds methyl acetate, Kokanai, Pape Georgiou, 1988, dihydrocarbone, Hendricks etal, 1976, 2,4, 8, 6p menthitrine 2,3 diol, Pino etal, 1999, and Pulgon, il idrisi, Belakdar, 1989. The difference in composition observed on the essential oils investigated is likely to be related to abiotic factors such as the climate specific to the regions of origin of the samples, geographical factors such as altitude and the nature of the soil. The results obtained show that all the bacteria tested are inhibited by the amount of essential oil used. The biological test gave us a strong sensitivity of Acantobacter baumannii with a diameter of 1.6 cm, followed by Salmonella, 1.4 cm. For Escherichia coli, Staphylococcus aureus, and Klebsiella pneumoniae showed approximately the same sensitivity. The first has an inhibition diameter of 1.1 cm, the second and the third both have an inhibition diameter of 1 cm. The inhibitory effect of Mentha Rotundifolia oil that has been recorded on Citrobacter freundii is 0.8 cm, whereas for Pseudomonas aeruginosa, the essential oil applied in the antimicrobial activity was not successful. Table 2. Table 3. Different concentrations of He and their effects on bacterial strains. The minimum inhibitory concentrations, MIC, of the Mentha Rotundifolia essential oil are determined according to the method reported by Rhaim etal, 1993, and Farah etal, 200I. The search for the minimum inhibitory concentration showed that the dilution of 1/200 is the minimum inhibitory limit. It is the lowest concentration which showed an effective antibacterial effect against the bacterial strains tested, namely Acinetobacter baumannii, 16 mm, Staphylococcus aureus, 10 mm. For Klebsiella pneumoniae, 10 mm, Salmonella suci, 14 mm, Escherichia coli, 11 mm, Proteus mirabilis, 56 mm, and Pseudomonas aeruginosa, 0. The first signs of inhibition started with the concentration of 1/100, which could block the development of these types of bacteria, and the total inhibition of all bacterial species did not is revealed only with a concentration of 1/50. Pour en finir par la déclaration que cette concentration est la CMI de l'huile essentielle de la plante Mentha Rotundifolia, Table 3. Conclusion. The chemical analysis of the essential oils of Mentha Rotundifolia by gas chromatography showed a strong diversification at the level of the chemical constituents with very variable contents in the plant. The chemical component, 2-isopropylidenecyclohexanone, is the most dominant with a rate of 11.99%, followed by eucarvone with a percentage of 11.42%, gamma-muroline with a percentage of 8.61%, 2-isopropyl 5-methyl-3-cyclohexane-1-1, 6.83%, and menthan 1,2,3 triol, 6.72%. The remaining substances found have a rate of less than 6%. The qualitative analysis of this essential oil of Mentha Rotundifolia has shown a great interest in the microbiological field by its inhibitory effect. The results obtained proved its bacteriostatic efficacy against all microorganisms tested with the exception of Pseudomonas aeruginosa. The minimum inhibitory concentration used is of the order of 1/200. The strain Proteus mirabilis was the most sensitive.